In drug research and development, the patient is the main focus. The mission for Novartis is to help the patient overcome a disease and improve their quality of life. The drug development process is designed to ensure that innovative new medicines are effective, safe, and available for patients in the shortest possible time. The first step in drug development is to discover the best targets for treating or preventing a disease. Targets are usually proteins in the patient's body, which are associated with a disease, or proteins in microorganisms causing a disease. The challenge is to identify which proteins are relevant and, more importantly, confirm their role in a disease. Increasingly, Novartis focuses on understanding cellular networks of proteins, or pathways. A single protein may transmit messages to several other proteins, sometimes in multiple pathways, affecting their function. Knowing how these pathways work and interact helps to identify the most appropriate target for a drug. The pathway approach allows Novartis to better understand the mechanisms of a disease. This knowledge, together with the desire to address unmet medical needs, determines the priorities in target discovery. In drug discovery, several methods like high-throughput screening and computer-based design are used to find chemical compounds or biologics that bind to the identified target. If a compound modulates the target in a way that is expected to alter the disease, this so-called hit will be refined to improve its safety and effectiveness, eventually becoming a drug candidate. Discovering and bringing one new drug to the market typically takes an average of 14 years of research and clinical development efforts and costs around $2 billion. Of 10,000 or more hits tested in early drug discovery, only one may eventually lead to a drug that reaches the market. In the late preclinical stage, further experiments are conducted on the drug candidate to ensure it is safe for patients and has the required pharmacokinetic properties, like appropriate absorption and metabolism by the human body. These experiments are executed with extraordinary diligence to minimize any risks to human test subjects. Animals play a critical role in the drug discovery process as well. Although much research and development can be done using various experiments or using computers, complex disease mechanisms can often only be understood through the use of animal studies. Also, governments and regulatory authorities require that medicines be tested in animals before they are tested in humans. Novartis keeps this research as limited as possible and always ensures that animal research is scientifically acceptable according to current standards and regulations. For these reasons, Novartis continues to use animals in its quest to find innovative, safe, and life-saving medicines for patients. At the same time, Novartis is committed to refining, reducing, and replacing the use of animals in research and upholding the highest standards in animal welfare. Clinical trial programs consist of several phases, each of which is focused on evaluating drug safety and effectiveness or efficacy. Information on ongoing clinical trials can be found at www.clinicaltrials.gov. In Phase 1 of clinical trials, the drug is usually tested in healthy volunteers to determine its safety and pharmacokinetics. In Phase 2, the drug is given to a group of about 100 to 250 patients with the disease to evaluate its efficacy and to determine the optimal dose. In addition, the safety of the drug and its side effects are evaluated, as these may be different in patients compared to the healthy volunteers tested in Phase 1. Novartis also conducts trials called Proof of Concept or POC trials, which are often a mix of Phase 1 and Phase 2 studies. A group of well-defined patients is treated with the drug candidate to provide an early indication whether it actually affects the target in the intended way or has a beneficial impact on the disease. These studies allow Novartis to rapidly invest resources on drug compounds, which are effective and safe. This way, the appropriate medicines can reach the patients faster. The data from Phase 1 and Phase 2 studies provide the scientific confidence necessary to continue the development of the compound in larger clinical trials. In Phase 3 of clinical trials, 1,000 to 3,000 or even more patients are recruited for research with the investigational drug. Phase 3 clinical trials are undertaken to confirm the effectiveness of the new drug, monitor side effects, compare it to established treatments, and gather additional information to allow the drug to be used safely. To bring a new medicine on the market, every new drug needs to be registered by the health authority. Data from all clinical trials are collected and compiled into a document called Registration Dossier. The Registration Dossier will include efficacy and safety data to support the final intended use of the drug. The dossier is then customized for different regions and countries around the world to meet the requirements of health authorities. This is a crucial step to ensure that patients all over the globe can access the innovative medicines that have made it through the complex drug discovery and development process. Once the registration dossier is approved and the product is launched, several activities will continue, like drug safety monitoring, which is mandatory. Post-authorization safety updates, annual reports, and any additional information required by the health authority must be provided at defined intervals as long as the drug remains on the market. Often, Phase 4 clinical trials are initiated to gather additional data not collected in the Phase 3 program. This may include efficacy, safety, additional benefit and risk information, as well as pharmacoeconomic data. Our mission is to provide safe and effective treatments that benefit patients and improve the quality of their lives.
Problem 3.6 says, a pharmaceutical manufacturer wants to investigate the bioactivity of a new drug. A completely randomized single-factor experiment was conducted with 3 dosage levels, and the results were the following. We have N observations and A levels. This is called the treatments. Therefore, the first step of solving Anova, the analysis of Breuil's problem, is making the first variable. We're going to make another column here called yi dot. This is the sum of all the values in the first row. Then we're going to add the same values of the second row, and then we're going to add the values of the third row, which are 100, 119, 147, 179. Then these results are going to be tested in the Anova table later on. So therefore, we are going to make another variable called yi double dot, which is the sum of these results. 445. And then we can start making the analysis of Breuil's table. The Anova table is stated like this. The first step is recognizing the treatments. The treatments are basically your A levels, which is in this case dosage, the amount of this drug that is supplied to the people, and we're gonna call them treatments. and then we're gonna have the error and then the total values. To complete this Anova table, we have to solve for these columns, which is the sum of squares the degrees of freedom then the mean square and then finally the f value. This is gonna be the one that is going to tell us whether or not the dosage in this problem is having an effect. So we're gonna make, for the treatments, we're gonna add these values. I'm gonna mark them with a dot. We're gonna square them like this. It's 119 squared plus 147 squared plus 179 squared. All of these are going to be divided by your n observations. This is observation number 1, observation number 2, 3, and 4. We're going to divide them into the number of observations, which is 4 observations. Then we're going to subtract the total value, which is 445 squared, divided by the total numbers, total tests that we have, the results, which is 12. 12 comes from 4 times the number of levels, which is 4 times 3. Then that's the first sum of squares of the treatment, which the result comes is 450.666 and so on. And then we're gonna find the total sum of squares. How do we get that? By adding each value squared minus the total value of the sums squared divided by the total number of results that we had in the experiment, which we can start like 24 squared plus 28 squared plus 37 squared plus 30 squared plus 37 squared and so on and so forth, minus, this is all the way up to 38 squared, minus, since it's the total value, we do not have to divide these by the number of observations like in the case of the treatments. We only have to subtract the sum of the columns. And the result will give us 738.916. It's important to keep in mind that we have to respect the significant numbers left from the point. And then, how do we get the error sum of squares? It's just a simple subtraction of the result coming from the total and then minus the result from the treatments, which is 738.196 minus 450.666. The result will give us 288.25. And then, what is the next step? The next step is easy. You have to recognize the degrees of freedom of each level, in this case the treatments. How many treatments do we have? The formula is basically the number of columns. You have to recognize which is the treatment and then find out how many levels are there in the experiment, and therefore once you have located them, you have to subtract the number of levels minus 1. Number of levels minus 1. Therefore, 3 minus 1 is 2. This can vary. You can have even 4 levels, and then comes 3, and many observations will give you another number here. It's depending of the experiment you run. Therefore, the total number, how do we get the degrees of freedom of the total? It's just easy. It's the same formula, but with all the data. All data minus 1. We have 4 times 4 columns times 3 levels give us 12 data. Minus 1 gives us 11. And it's the same operation as we did in the sum of squares. To get the degrees of freedom of the error, we have to subtract 11 minus 2 gives us 9. Then how do we get the mean squares? The mean squares are basically the division of the sum of squares between the degrees of freedom. In the case of the treatments, mean square of treatments equals 450.666 divided by 2. That's the degrees of freedom. The result is 225.33. That's the result. That's the mean square of treatment. The next step is finding the mean square of the error. It's the same operation as we did in the treatment. Therefore, it's the mean square of the error. That is 288.25 divided by its respective degrees of freedom, which in this case is 9. The result is 32.027, and that's the result. And the next step, in order to determine if what is going to be the value that we're going to have to compare with the tables. If tables have a specific value, and then you have to find it according to the degrees of freedom and the percentage of liberty that you have in your experiment. In this case, since the problem does not specify the percentage, we're going to have to assume it's 0.05. But let's return to the F calculated. To get this number, we have to divide 225 0.333 divided by 32.027. This will give us the the F value of the experiment, which is 7.035. There's the Anova table. It is complete, but that's not everything the problem asks us to do. This is just the first step. Now, what comes first? We have to determine whether or not the hypothesis is rejected or is accepted. If we accept the null hypothesis, or we accept the alternative hypothesis, and therefore we have to recognize that we need an F table that has already the values from F. In this case, we're gonna look up for 0.05, which is the percentage that we are assuming, of course, where the problem can specify 0.01 or 0.05, most likely. And then we're gonna use the degrees of freedom of the error. In this case, is 9. Always, to find the value of F in the tables, we have to use the degrees of freedom of the error. And that's the number we're going to compare with the one we just got. As I was saying, we need to find the F value in the tables, which is, in this case, 0.05 is the percentage. Then we have the levels, the degrees of freedom of the treatment, which is in this case 2. And then we have the degrees of freedom of the error, which is, in this case, is 9. Then we go to the tables and we go F table, F distribution table, and then in the x-axis we're gonna look for this first number, and in the y-axis we're gonna look for this number. The result is 4.26 in the F distribution table. We have to compare this value with this value. If the value we calculated is higher than the one from the tables, then we reject the null hypothesis. H 0 rejected. That's the first step of solving this problem. This is called the question is basically, is there evidence to indicate that dosage level affects bioactivity? In this case, it does. Then the next step of solving this problem, what the problem is asking us is analyze the residuals from the experiment and comment on the model adequacy. How do we do this comparison of the residuals? It's plain easy. We have to add another column here of the average of these values for every row. Y average. It's basically just calculating the average of the row, as I mentioned before. The values are 29.75, 36, and 44.75. Once we have these numbers, it's important we use them in order to obtain the values of the residuals. What are the residuals? It's just simple formula. We have this number which is 24 minus the average, which in this case is 29.75. We will have to do every single value of the row which is with its corresponding average in order to get the residuals. In this case, the first number, this actually gives us minus 5.75. If we do the same, 28 minus 29.75, we will get 1.75. 37 minus 29.79 will give us 7.25. And the last value is 30 minus 29.75, which is 0.25. We'll have to do the same thing with the other two rows. For example, 37 minus its corresponding average, 36.75, which is 0.25. Then we'll just have to keep moving forward and doing the same operations. I'm just going to keep writing the results from the operation as I did before. I already have made the calculations, and in this case we're going to do the same thing. 42 minus its respective average, 44.75, and then we're going to do the same thing for the column. 2.25, 7.25 minus 6.75. There we go, and we have the residuals. What's next? We have to make sure the data successfully approves the normality test. How do we do a normality test with the residuals? This is what the problem is asking us again. It's depending whether or not the problem asks us to do this procedure. In this case, we're going to make an order. This is just a probability and normality test. We have 12 data, so we're going to have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, and 12 values. Then we are gonna write the result of the residuals, these numbers only. Those are the numbers that are gonna be up here in this table. Then the next column is gonna be called probability. It's just a simple formula. It is the number of order minus 0.5 divided by n, which is the total amount of data that we have, which is 12 in this case. So, how do we reorganize the results from the residuals in this table? It's it's just easy. We have to order them from minimum to maximum, from the biggest negative to the biggest positive. I already have made the process of seeing which one is the smallest number and which one is the biggest number. I'm just going to write them down. You can check them with your results later. There we go. That's the proper order of the residuals that we got in the step before. Now, we have to make this operation. The residual is just to recognize if it's a normality test that is the one that follows this experiment. And therefore, we are going to have to make the number order. I'm just going to make one example and then we're going to write them the results, because I've already done the operation, so I'm just going to write just the first. So it's 1 minus 0.5 divided by 12. The result gives us 0.041. This is just the probability. And then we are going to have to make this exact same operation all over with the number of order that we have. This can be 2, 3, 4, 5, 6, 7. These values are constant. This is the one that it's variable. I'm gonna write the result now. Mistake. Sorry. How do we know that we got actually How do we know that we actually can be right and we're doing the right process? It's easy. The probability is a percentage. And you have already transposed the residuals from smallest to biggest. Therefore, the values from the probability are going to be from smallest to biggest. This is going to tend up to 100. What I mean by this, it's not always going to be 100. The sum will never be 100. It's just a perspective that it's increasing the percentage. Then we're going to have to make a probability test table that is specified, and we're going to check for normality test. This is the second part of what the problem is asking us to do. That's the residuals table, but it also asks us to compare the residuals with the fitted values. The fitted values are these ones. The average we got long time ago. Therefore, we have to do the same procedure as we did, but it's a completely different table. What I mean by this is we're going to have to make a simple subtraction and transpose the values from the residuals we got. In this case, I'm gonna write down the average we got in the step above, right here. So we have 29.75, 29.75, 29.75, 29.75, which is the first value. Then we're gonna have to relate the residuals to this number. So we're gonna have four columns, and therefore we're gonna write down these four values. And we're gonna do the same with the same. We're gonna do the same with the next averages. And we're going to write down, transpose the values we got from the residuals. I'm sorry, the handwriting is not that perfect. Next up, we're gonna make a table. I'm gonna make a really fast sketch from here to conclude this problem. In this case, we're gonna have in the x-axis the average. We're gonna have to make an interval, since we do not have specific values, and we're gonna try to make as even as possible the table. So we're gonna have 30, 35, 40, and 45, which are the range, sort of the average we got here. And in the y value, we're gonna have the residuals. As you can see, the minimum is minus 6.75. But as I mentioned before, to make an even table, we're gonna have to make a range and intervals throughout the y-axis. In this case, I'm gonna go from minus 5 up to the middle, which is 0, and then plus 5. I know I have just one value down here, but obviously this is minus, I'm sorry. But we're gonna trace the dots so we can figure out whether or not the residuals, how the residuals are compared to the fitted values. I'm gonna trace a dot line here so we have a reference. I'm gonna draw simple points of the residuals in their value, and afterwards we're gonna determine how distanced are from the average in each observation. This case is minus 5.75, which is down here, minus 1.75, which is around here, 7.25, which is over up here, and then 0.25, which is around here, give or take. Then we're gonna In this case it's not 30, it's 29.75, but my scale is too small to make that specific, and this way I can make sure they're on the same line. I'm gonna do the same thing with the rest of the averages. In this case, follows 36.75, which is around here. So we have 0.25, 7.25, minus 5.75, minus 1.75. Sorry, 0.25 is up here, minus 1.75 is down here. So, and the next is 44.75, which is over here approximately. Then we have minus 0.75, 2.75, which is over here. Let's make a guess. Minus 2.225. And then plus 7.25. And then minus 6.75. It's over here. Then, what's up next? I'm sorry I messed up. This is a positive sign, since we have minus positive. This one should be positive. This dot should have never been there. As you can see, the dots are basically in the same levels. So we can conclude one thing: there's nothing too unusual about the residuals. That is, this experiment is basically not worth doing. I'm gonna leave this right here as a final image of the procedure. Thank you very much, and hope you can do more problems on your own. Thank you.